And thinking about big trees and mammals, kind of the first thing that comes to my mind at least is this Emmons super tree from 2007. And this was a really important tree to push the field forward. It is a combination of 2,500 source trees, both morphological and DNA. A lot of these are cytochrome B trees at the family level that have then been composited. So you get some interesting properties of polytomies throughout the tree. It's been heavily used though. If we look a little closer at the tree, these polytomies really pop out. So looking in the rodents here, you get these giant combs at basal nodes, same in the bats. It actually ends up being over 50% unresolved nodes or polytomies. And so the problem with that is that functionally, when we do analyses of tempo and mode on a tree like this, there's the hard polytomies and soft polytomies are confounded. So whereas we might have situations of biologically rapid radiation where data can't really resolve it, there hasn't been substitutions, that is confused with situations where we just don't have any data. So that ends up being pretty troublesome when people are analyzing evolutionary rates on this type of tree. And so there's been improvements since 2007, first in updating the taxonomy from the 93 mammal species of the world to the 2005, and then in resolving the polytomies using a birth death model. But you can see that the structure of the tree really doesn't change very much. These are all very short nodes at all the polytomies, so they're still inferred as areas of rapid radiation. Most recently, there was this Faubrian spinning super tree that is certainly an improvement. It's based on small DNA trees at the genus or family level. About 58% have DNA, but it's still doing this kind of random polytomy resolving and lots of manual rescaling of branches according to different studies. So really what we want to do is move past this super tree approach for mammals and build a large DNA supermatrix. So that's what I set out to do here, really with the goal of having more accurate branch lengths for looking at questions of tempo and mode in this group, and to really do so across a posterior distribution of trees. So I think that kind of summarizing this, super trees are certainly useful, but for different types of questions than a supermatrix type tree. So looking at consensus levels of support for evolutionary relationships, both at deep scales and at the species level, super trees can be useful, and in particular, reconstructing how traits evolved. But then, when we're interested in questions of how major radiations began and in relation to what events or rates of speciation and extinction, we really need more accurate branch lengths. And so, I'll talk a little bit first about how we assembled this large DNA supermatrix, and then this two-step tree building approach of using a backbone and patch plates that was first developed in the bird tree of 2012 by my supervisor, Walter Yates. And then talking about some analyses of diversification rates on this tree. But first, thinking about how we got this DNA data, because it actually is not trivial. So the way we did this was to use a blast-based approach and gather data from GenBank. So we assembled what I'm calling DNA weights. So per gene, trying to cover the diversity kind of representatives for each mammalian family. So we started with the DNA data set of Meredith et al., 2011, which had 27 nuclear genes, and then I added 4 mitochondrial genes from GenBank, also covering sort of the family level diversity here. And then those are used to blast against a local copy of GenBank that's subset to just mammalian sequences to return all possible homologous sequences for each gene. That includes the taxonomic information too. So as you can imagine, there's lots of redundancy when you do something like that. And so we did some filtering and cutting of redundancy and batch to end up with these unique longest sequence per gene per NCBI taxon ID, which then creates a separate challenge because then we need to translate NCBI taxonomy into something that is accepted for mammals. So to do that, I kind of ended up matching to IUCN 2008 taxonomy, which is approximately similar to the 2005 mammal species of the world, including a synonym checking, but we still had about 750 names that we had to manually look up. And actually, this ended up being an important process because there's lots of genetic data actually on GenBank for species that have been described since 2008. So we added close to 400 species to this taxonomy, and ended up with this kind of master taxonomic list of close to 6,000 species, including 100 that are recently extinct, about half of DNA, and 13 domestic species that are, in every case, they're sampled for all of our genes, so they're useful markers. And so Oldfield Thomas, if anyone knows, described about 2,000 species of mammals himself, and so we think he would be happy with this sort of effort to attention to detail of taxonomy. So we still have, we got sampling for close to 70% of the species, but lots of junk still coming off of GenBank. So we did this kind of iterative approach of aligning genes, using this different software, looking for stop codons and misalignments, building gene trees, looking for rogue taxa from these bootstrap samples, and examining misidentified sequences, things that are identified as rats but come out in primates, things like that happen a lot. So we ended up throwing out close to 2,000 sequences, and we ended up with this final sampling of 4,098 mammal species for greater than 1 gene, or equal to 1 gene. This is what the big supermatrix ends up looking like. As you can imagine, it's very sparse, but certain genes, this is cytochrome B, that's CO1, these are some of the nuclear exons like GHR, are quite heavily sampled, and then for a few taxa, or about 60 taxa, are sampled across the whole alignment. So we're calling these type 1 species, and with these DNA sampled species, we're building stage 1 trees with Raxonal, kind of for the topological constraints, so we're collapsing nodes that are less than 90% bootstrap supported, and then using those topological constraints in Bayesian framework in Mr. Bayes, where we also add in missing species. So, if anyone's familiar, this is called the pastis method. These missing species, so the 1812 species in our taxonomy that we didn't have any DNA for, are broken down into things where there's a congener sample, so we constrain that to the genus, or where there's a confamilial sample, we constrain that to the family, or in 8 species cases, we actually have 8 species from 3 extinct families that are not, we don't have any DNA for. But actually soon there will be a thylacine genome, so we can add that in. So this is kind of what that ends up looking like. We get these stage 1 constraint trees, stage 2 Mr. Bayes posterior samples that include these imputed species kind of within taxonomic constraints. And then, this is what we did this across 27 patch clades. And I don't expect you to be able to read all this, but the largest patch clade was Muridae and Crucidae, these 2 rodent families that are very speciose. These end up being hard monophyly constraints. So we didn't want to have a ton of patch clades. We wanted to be inferring that topological uncertainty throughout the tree. But then these patch clades are rescaled to the outgroup divergence time within a backbone analysis. So this is where we're incorporating the temporal information. I did both tip-dated approach on the backbone and node-dated approach. And this ends up all being stitched together to get these full trees in a posterior sample. So here's what the backbones end up looking like. The tip-dated is pretty interesting. We use this Mesozoic mammal matrix out of Gigi Luo's lab at Chicago, and it ends up having very few assumptions about placement of fossils, which is nice. The morphological data places the fossils, and then we only have the stratigraphic information. And so in relation to the KPGs is a good kind of visual look at the branching here. The crown of placental is at 102 million, which is a little earlier than some people think, but it seems somewhat reasonable. In comparison, we did this node dating approach as well with 17 calibrations from the Benton compilation that covers tetrapods. We picked out these 17 for mammals. And the difference here is allowing for a soft maximum age, where the tail of the exponential, the 5%, can be older, versus a hard maximum where it's just a uniform cutoff. You can see the difference in age here with the crown and placentals, and in relation to some of the early branching. So we're going to be producing trees that are based on each of these three backbones. Looking at, this is the full thing stitched together, right? So it ends up being sort of dramatic looking. The branches here are colored by diversification rate, this DR metric. And this is the median across 10,000 trees of the posterior sample. And so if you're not familiar with this DR metric, it's basically a way to look at the amount of shared branches. It's the amount of shared branch length and in relation to the number of downstream species. So it's sort of a look at the splitting rate from root to tip. So high DR, these red values are short branches shared by many species, and low DR are these long depopulated branches. So you can see the distribution of DR across the tree. And if you're curious about what this looks like when we prune out all those imputed species, so this is pruning those out and then recalculating DR. You get the same, I'll go back, the same sort of patterns with, this is the primates here showing up as hot. Similarly here, they're still showing up as hot. And this is the comparison with the distribution of branch length, of DR, sorry. So as you would expect, pruning out lots of species lowers the diversification rate, so you get more of a peak at the lower diversification rates across those. If we zoom in, it's pretty interesting to look at in a little more detail throughout this. Primates here, so high rates of diversification, except for this extinct sloth lemur lineage, which is pretty interesting. We were able to add this based on a few mitochondrial genes that were on GenBank. Looking at our clade Homo sapiens here with the three extinct other members of our genus, also showing up hot. Jumping over into the Cetartiodactyla, dolphins have these fast rates of evolution. This is a result that's been found by Dan Roboski's group and others. Tucos over in the rodents. So there's some South American rodents that I studied for my dissertation. We found a similar result, so that's actually really heartening. And looking at the murid rodents, Radis comes out as having particularly fast rates. So there's a lot of information here. We're just sort of scratching the surface. But in conclusion, we were able to put together this big supermatrix for mammals, have much more accurate branch lengths, and really a consistent framework across the whole clade for asking evolutionary questions, in particular all these parallel radiations. And looking at this in space is going to be quite interesting also. I'll just mention this is kind of the first version of this tree. We're adding ultra-conserved element sequencing for a lot of basal clades as well as unsampled lineages as part of this NSF BirdLife grant that I'm doing my postdoc on. And so I'd just like to thank these people, these collaborators on this grant, people in our lab, and I'll take any questions. Thanks.